On this week's Arabidopsis Research Roundup, I'm delighted to be joined by Lars Ostergaard from the John Innes Centre. And we're going to be talking about a paper that's been out about a month now in Genes and Development with the title of A Non-Canonical Auxin-Sensing Mechanism is Required for Organ Morphogenesis in Arabidopsis. So thanks very much, Lars, for joining us today, and it'd be great if you could give us a little background and then describe what you did in this paper.
Thank you very much, Gary. Over the last maybe 10 years, a general mechanism for how hormone signaling is occurring in plants has emerged. And it seems like a common theme is that many hormones are signaling via degradation of specific transcriptional repressors. This is true, for instance, for gibberellin, for jasmonic acid, and also for auxin. And in a canonical auxin signaling mechanism, it is specifically the auxin response factors, the so-called ARFs, that mediate the auxin response and activate auxin responsive genes. These are, in the absence of auxin, inhibited by some repressors of transcription called auxIAA proteins. Yeah. And when auxin becomes present, these proteins are degraded via the 26S proteasome. Okay. They interact with an F-box protein called TO1, or a family called AFB of similar kind of proteins, and then they become degraded, and that releases the repression of the auxin response factors. Okay. So in Arabidopsis there are 23 of those, and they all have a specific domain that allows them to interact with these auxIAAs, except very few. One of them is called ARF3, or etin, as it's called in developmental biology. And etin does not have this domain. It cannot interact with these auxIAA repressors. So therefore, auxin signaling via etin must be mediated via a different pathway. Okay. So we found a role of etin in gynoecium development, or rather, this has been known for quite some time when the mutant was identified that it has some severe defects in gynoecium development, which is the female reproductive tissue or organ in the Arabidopsis that produces the ovules and eventually the seeds. So in etin, there are some clear defects in, for instance, polarity establishment. Okay. And we found that etin interacts with another protein in establishing the gynoecium, which is called indehiscent. Okay. So these proteins interact with each other, and they interact with a domain that etin has, which none of the other ARFs have, and it has it instead of this auxIAA domain. And what we found was very exciting was that the interaction between etin and indehiscent was dependent on auxin, or rather, auxin seems to disrupt the interaction between the two, and that's important for the way that they regulate downstream targets. So we could, for instance, make a version of etin and a version of indi which no longer sensed auxin. So this way of signaling auxin would no longer occur, and that led to some also severe defects in gynoecium development that were different from the one in the etin loss of function. One of these target genes specifically, which seems particularly relevant, is a gene called pinoid. And pinoid encodes a protein kinase that regulates the phosphorylation state of pin proteins, which are auxin efflux carriers. So they transport auxin out of the cells. They often do that in a polar manner, which is very important for the action of auxin, that it's polarly distributed. Pinoid makes sure that this polarity is established, so that pins will only be located to one side of the cell and transport auxin in that direction. If pinoid is repressed, then the pins are located to all sides of the cells. And there is a specific time point in the gynoecium development where this is important, and this is in the establishment of radial symmetry at the apical end of the gynoecium. Okay. If pins are not apolarly oriented there, then the gynoecium would be split. It would be bilaterally symmetric, so you would have a split at the top, and fertility would be severely reduced. Okay. So we found that in the absence of auxin, etin and indi together will repress pinoid to make sure that we get this apolar distribution of auxin. Right. But then, as auxin accumulates, which it does at this site, then the two proteins, etin and MD, change from being repressors to becoming activators of penoid. Mm-hmm. And this is important for the following developmental stages of gynoecium development.
Okay, I see. So there's these two phases of action depending on the auxin concentration, which then disrupts this interaction. So you explained about the FBOX proteins and the auxin proteins, and that signaling module is dependent on degradation. Do you have any evidence that the indehiscent protein is degraded after the auxin interaction?
No, we don't have. In fact, it seems like it is most certainly still present and not degraded. It's resistant to degradation. Okay. So what we find is that if we treat gynecia with auxin, mm-hmm. and we then do a chip assay, so we have an etin protein fused to a GFP mm-hmm. that we know is functional because it can complement the mutant, then we can pull down this particular protein after treatment with auxin, and it's still present. It is still bound to the same targets, but it just seems to have altered its activity towards those targets from being repressor to activate. It seems to be a degradation-independent signaling.
Okay, so a totally different paradigm, really, in the way that auxin signaling occurs. That's very exciting.
Yes.
Yeah. So you've talked about the gynoecium development so far, but in the paper you also show some evidence that etin is not only active in that tissue, but elsewhere in the plant as well.
Yes. So we did a screen with a yeast 2 hybrid screen with etin against a library that contains all transcription factors from Arabidopsis that was made by the Regia consortium some years ago. Mm-hmm. And where we found there was that etin could interact in a similar auxin-sensitive manner with other transcription factors that were not related at all to indehiscent. Indehiscent, by the way, is a basic helix to helix type transcription factor. But we also found homeobox domain transcription factors that interacted with etin, plethora family, Canardi family members, and so forth. Mm-hmm. And what we found was that when we have this insensitive version of etin that could no longer sense the auxin, it's just one point mutation, rather 2 point mutations, that makes the protein unable to sense auxin anymore. That has consequences for other parts of plant development. Okay. And always in places where auxin maxima would emerge. Right. So what we expect is happening is that in a similar manner to the gynoecium and the indehiscent, etin will interact with tissue-specific transcription factors in other places and mediate this kind of change in polarity. So we think this module is particularly well suited for inducing changes in polarity.
I mean, there's a lot published about the other signaling module that you described at the start. So how do you think, do you have any idea about how this module, this etin, indehiscent and etin and the other transcription factors, how that interacts with the other signaling module? Is there any interaction between them? Because we know that those other proteins play a role in many of the developmental processes that you've talked about. So how do they interact within those processes?
That's a very interesting point. Great. And in fact, so what we could imagine happening, and this is based on work from, for instance, Dolph Wyers's lab, that ARF proteins can also dimerize via their DNA binding domain. Okay. So it is possible that etin, which has this etin-specific domain at the C-terminal that seems to be responsible for this novel pathway, but it also has a DNA binding domain which is very similar to other auxin response factors. So it is possible that dimerization can happen at the DNA level between etin and maybe a more canonical factor. In this way, they are made. This other way might be this other ARF might be repressed in the canonical fashion.
Okay, so that is something I'm sure that you're looking at the moment.
Yes, that's right. So we would like to, of course, test if this is the case.
Okay. So my next question, just as we get towards the end here, might be a bit more of a deeper auxin question, perhaps. So we know that the FBOX auxin interaction. That the auxins can be degraded by numerous different types of auxin, so 2,4-D and NAA, which are synthetic auxins. But you show in this paper that it's very specific for IAA, so indole 3-acetic acid. So does that suggest that this might be a tighter regulation or a tighter interaction, and that the other interaction maybe is a bit more fluid because the binding site might allow kind of different auxins to get in there. I don't know what do you think that means?
So I agree with you. It does suggest a more tight regulation, a higher level of specificity, certainly in the interaction. We don't know at this point where IAA comes in to the equation physically, which protein does it interact with, in what way does it interact. Mm-hmm. We don't know that yet. They are trying very hard to make protein of auxin and of some of its partners to be able to obtain a crystal structure and see what effect that might have. And also simply just doing some basic biochemistry, finding out what the KD association constants are for this interaction. That's what we will be very interested in, but we have really big difficulties in producing the proteins. So we haven't been able to make any biochemistry yet about any potential interactions. We are not claiming this to be an auxin receptor.
Yeah, I see. No, it's fascinating to find out because you can imagine within the cell, this ARF protein is likely within the nucleus, and the site of auxin perception elsewhere is a little uncertain. So whether this is the first perception or it follows on, and if it's tighter than the other perception, so there's lots of questions to be answered. Certainly, that's for sure.
Absolutely, yes. Yeah, indeed. One exciting thing that we are moving towards right now is to see what the influence is on the chromatin dynamics.
Okay.
Because there's a very interesting analogy between the way that this mechanism is and the way that, for instance, the thyroid hormone receptor works in the animal development. Okay. The thyroid hormone is constitutively bound to its target genes, but without the hormone, without the ligand, they recruit chromatin repressors.
Okay.
That would then repress expression of the genes that it's targeting in the absence of the ligand. But when the ligand becomes present, it changes its conformation. And then it recruits activators of transcription, so chromatin remodelers that will allow transcription to take place.
Okay.
And it could be a similar mechanism here between hormone dynamics in animal systems related to this particular pathway. Mm-hmm. So that's something we also are trying to follow up on.
Well, it certainly sounds like a very exciting time in the lab with this biochemistry and now this new development. So thanks very much for talking us through this paper. It's been really interesting. Thanks very much, Lars.
Thanks very much. Good night.